Okay, good morning and welcome to Let's Crack NEET UG English. Okay, let us first check the audio part, if the audio is clear or not. Just message me if the audio is clear. And what we are going to be doing today is a recap of the non-cyclic photophosphorylation, which we have studied in the previous lecture, and then we will start with cyclic photophosphorylation and dark reaction. So is there any kind of an audio problem? Good morning, Rahul. First of all, I want to tell you something, that there is some construction work going on in the office next to mine. So maybe, we might find little disturbance today, but we'll try to maintain the audio as best as possible. And if in case there is some kind of a disturbance, just message me, so I will repeat that particular part. Okay, so far you have written that audio is good, so that's fine. Now what we will do is we'll have a quick recap of non-cyclic photophosphorylation. This is the process which we studied in detail in the previous lecture. And after the recap, you can ask if there are doubts. I'm sure you have utilized Sunday to go over that non-cyclic photophosphorylation and the other things which we did in photosynthesis. Okay, so let us go over the basic things which we do every day, and this is the schedule for our channel, that is Let's Crack NEET UG English, and we are doing this chapter of photosynthesis in plant physiology. NEET UG, that is Let's Crack NEET UG, this is another channel where it is the lectures are Hindi English mix. There are only 2 lectures left on this channel, and those are going to be on human physiology, human heart, and the functioning. And this is another channel, that is Biology 101, and here I have started human physiology, digestion and absorption. So we will continue with this chapter, and this is going to take some time, so you can join this channel also. The previous lectures are also available here. Right now we are on this channel, which is Let's Crack NEET UG English, and if you have joined me once, there is a question, let me take that. Rahul is asking, ma'am, once you start plus classes, would you stop this free class? See, there is a division. There are free classes which are going to be conducted maybe on the YouTube channel or only on the app. On app, there are 2 types of classes: simple special classes and then the plus classes. The special classes which I'm taking right now, where I'm talking about genetics, those are free. But once I'm on plus, then that is definitely going to be a paid one, and that is why you need to install this app. So once you install this app, you would get access to 2 things. The app is Unacademy learning app. So from your app store, you can install it. There are 2 categories. One is simple special classes, which are free, and then the plus classes, which is actually a structured program, keeping in mind NEET 2021 or NEET 2022. And once you're on the app, you would get all this live classes from various faculty members who are experienced for training students who are appearing for NEET. So all those lectures will also be available to you. Now this is the subscription plan which I was talking about. And for your your convenience, the plan has been designed in such a manner that you can choose just a month plan so that you know how exactly it works, the poll, the quizzes on a daily basis, how these things take place. And if you find that everything is good, physics, chemistry, biology, all those lectures are good for you, then you can go for a long-term plan. But going for a long-term plan is beneficial financially as well as because you now get to know the teacher and the teaching methodology and all those things. And this is the code which you need to put in whenever you take the subscription so that you get 10% discount. So this is about the subscription plan. Now let us come to non-cyclic photophosphorylation, a quick recap, and after that I'll give you time to ask doubts in case if you have. Now in this non-cyclic photophosphorylation, what we talked about was that this is a major pathway, and in this major pathway there were two things which were synthesized. We get ATP and the reducing power. Now in this same non-cyclic photophosphorylation, the reaction or the pathway starts with PS2. When sunlight falls on these chlorophyll molecules, a pair of electrons gets excited. It is accepted by its primary electron acceptor. Now this electron, which is at a higher energy level, is going to come down the gradient through electron acceptors. There were three: plastoquinone, cytochrome complex and plastocyanin. And finally the electron was sent to the chlorophyll of photosystem 1. So here there is this pair of electron which is moving from higher energy level to lower. Now when it is moving down the gradient at one place, that is at cytochrome, it releases that extra energy, and that energy is used to add this inorganic phosphate to adenosine diphosphate, and that is how this ATP is synthesized, and the electron has come to PS1. PS1 also receives photons. So from here also a pair of electrons gets excited, and this is accepted by its primary electron acceptor, that is primary electron acceptor of PS1, and this was primary electron acceptor of PS2. Hema is asking a question about the class. Hema, just let us go over this recap thing, and then we will talk about these things, okay? By the time I'm done with this recap and you start answering doubts, at that time I'll take some more questions that you have, okay? So the electron now from PS1 and its primary electron acceptor is given to NADP. So this NADP has received the pair of electron. Now let us come back to this PS2. This PS2 has lost a pair of electron. And from where do you think this electron pair is coming? It is coming from some atom. And if an atom loses an electron or electrons, then that atom would change into an ion. And we know that ions and atoms have different properties. So the properties of our chlorophyll molecule are going to change. So there is an important thing which is to be done here, that is replacing those lost electrons. And for that, water undergoes splitting in presence of sunlight. This process is known as photolysis. And for this, what is required is Mn. Mn is required for this photolysis. When water splits, a pair of electrons, a pair of protons, and half O2 are released. So now this pair of protons is given to this photosystem 2. So the loss has been made up. From photosystem 2, a pair of electron was lost. So now this pair of electron is given back to that, so that the structure of chlorophyll molecule is maintained and its property remains the same. This pair of proton which is produced is given to this NADP. So NADP has received 2 electrons and 2 protons, and now it changes into NADPH2, which is also written as NADPH plus and H plus. Either ways it is correct, but exactly it is like this, but we can write it as NADPH2. So in non-cyclic photophosphorylation, the electron which is lost from PS2 never comes back to PS2, and that is why this pathway is called non-cyclic. Then we called it photo because the sunlight is essential for this. And then there was a word which was phosphorylation. Phosphorylation is addition of phosphate. Now 2 things are produced, ATP and reducing power. This is our reducing power, and these two things will be used up in dark reaction. Okay, so this is a recap of non-cyclic photophosphorylation. Now let me take your questions, and in case if you have doubt, just start messaging those questions. Priya, good morning. Okay, Kalai has also joined, Khalid has also joined. Okay, now there was one question. Okay, ma'am, this is Hema's question. Ma'am, this free classes don't have any plan. They do, but it is like actually we are taking chapter by chapter. So if I have started one chapter, obviously once I'm finished with that chapter, I'm going to take up the next. But yes, if you're talking about structured thing, then that is only on the app in special classes. Okay, I'm waiting for the questions in case if you have any doubt. Mesophyll cells in C3 and C4. Jayant has a question on this. Mesophyll cells in C3 plants have chloroplast, which are granule, and that is why light reaction, dark reaction both takes place in mesophyll cells. We will talk about C4 plants in some maybe next lecture. We will be talking about C4 cycle. C3 cycle we will discuss in today's lecture. So C3, C4 both have mesophyll cells, but in C3 mesophyll cells have granule chloroplast, and in case of C4 they have granule, but agranule chloroplast are present in bundle sheet cells. Okay, so far nobody has asked any doubt. I guess the first part that we did, non-cyclic photophosphorylation and the things before that, are absolutely clear to you. And don't forget, towards the end of this chapter we are going to have a quiz. So if you have understood, you should be able to answer all those questions correctly. Okay, so shall we start with the next pathway, that is cyclic photophosphorylation. Okay, Kalai has said yes, Rahul says yes, so I am going to go ahead with the next pathway, and once we are done with the next pathway, then again, okay, Jayant is asking about cytochrome complex. See, this is the cytochrome complex. This is the place where ATP synthesis takes place, and we said this ATP synthesis at cytochrome is known as chemiosmosis. And this is a very, very, very important thing. And chemiosmosis we will be talking about later on, maybe in the next 2 lectures in detail. Alright, so we are on the second pathway now. And why we are calling it second pathway? Because in this pathway, the substances or the products which are obtained are not like everything which is required for dark reaction, and that is why we said that it is like only 20 to 15% times this pathway takes place. Majority 80 to 85 is non-cyclic. Now, as the name is cyclic, what indication does it give us? Why did we call the earlier pathway non-cyclic? The reason was the electron pair which is lost from a photosystem never comes back to the same photosystem. And when we are saying cyclic, so the hint is that the electron which is lost from that particular photosystem must be coming back to the photosystem. So now here let us draw the pathway, and I'm going to draw this reaction center chlorophyll, and around the reaction center chlorophyll there are other accessory pigments, and this is PS1. Now these things will also help you understand the differences between cyclic and non-cyclic photophosphorylation. So this is PS1. That means the reaction center chlorophyll pigment is P700. In non-cyclic, both the photosystems were involved and the pathway starts with PS2. Whereas in case of cyclic, there is only one photosystem involved and the reaction is going to start with PS1. And this photosystem has its primary electron acceptor. Alright, so now when sunlight falls on this photosystem, then what must be happening? Sunlight falls here and these pigments must be absorbing their photons. So depending upon its capacity, it absorbs the photons and passes on those photons to the reaction center chlorophyll. When sufficient energy is available to the reaction center chlorophyll, a pair of electrons would get excited. And this pair of electron will be accepted by the primary electron acceptor of PS1. And now this electron which is at higher energy level will be brought back to PS1 through an electron transport chain. And now you know why we call it cyclic photophosphorylation. So this pair of electron which was excited or which leaves photosystem 1 comes back to photosystem 1, and that is why we are calling it cyclic. It is photo because light is required, and there is phosphorylation. Now when this electron comes down the gradient from higher energy level to lower energy level, again this is FD. FD stands for ferridoxin. CY stands for cytochrome complex, where ATP is synthesized, and then there is plastocyanin. So here is PC. So now when the electron is passing through cytochrome, the energy is released, and this energy will be used to add inorganic phosphate to ADP, and what will be synthesized is ATP. Now in this process, in this pathway, only one photosystem is involved and the electron is coming back to the same photosystem. So do we need photolysis of water? So here, if we have to write the important points, only one photosystem is involved, and that is PS1. Second is, as electron pair comes back to PS1, no photolysis required. And if there is no photolysis, then will oxygen be given out as a byproduct? As no photolysis, no oxygen is given out. So in non-cyclic photophosphorylation, because the electron doesn't come back, you have to, or plant has to undergo this process of photolysis. Because water is splitting, oxygen is given out as a byproduct. In case of cyclic, because the electron comes back, so you don't have to make up that loss because there is no loss. So in that case, no photolysis, and if water doesn't split, no electron pair, no proton and no oxygen. And if no proton means no reducing power. So here there is only ATP synthesized, no reducing power. No reducing power is generated. See, this is why this pathway is not the major pathway, because for dark reaction to take place, two things are required. Because in dark reaction, carbon dioxide is reduced to obtain carbohydrate. And for reduction, reducing power is required, ATP is required. So for dark reaction, ATP and reducing power are required, both the things. So if only cyclic photophosphorylation if that takes place, then only ATP would be synthesized. That means the requirement for dark reaction is not fulfilled. And that is why non-cyclic photophosphorylation is the major pathway, because it gives us ATP as well as reducing power, but cyclic also takes place, and there is only PS1 which is required. Now, do you remember the location of PS1? Where do you think the PS1 is located? This we talked about when we were talking about chlorophyll and the site of photosynthesis. So do you remember where PS1 is located? Can anybody answer this question? Location of PS1. We have talked about this in the earlier lectures. Okay, I'm still waiting for the answer. Location of PS1. Rahul has answered non-appraised part of thylakoid. Kalai has also answered as non-appraised part. One more location. This is correct. It is non-appraised membrane of thylakoid. So this is correct. Reshma, Rahul and Kalai have given the correct answer, but there is one more location. There is one more membrane where this PS1 is present. No, it is not stroma. Amazing creativity has answered at pigment has to be in the membrane. Stroma is the jelly-like material, matrix. Kalai has answered stroma lamellae. Okay, it is stroma lamellae. So now you know where PS2 is located. PS2 was in the appressed part and PS1 is in the thylakoid membrane, that is non-appressed part, and in stroma lamellae. Very good. So those who have answered, that means they have attended the earlier lectures and they are revising also. The others have not answered, so I guess probably there can be only two reasons. One, maybe they have not attended those earlier lectures, or the second is they have not revised. So such type of questions are asked in your competitive exams, and that is why you need to revise those things. Okay, so is cyclic photophosphorylation clear, and can we distinguish between cyclic and non-cyclic? If you have any doubt in cyclic, you can ask that. So I'm going to wait for a couple of minutes. Aradhya has also answered as stroma lamella. When will the next chapter start? As soon as photosynthesis is completed. So once we complete photosynthesis, we'll start the next chapter, and I'll give you the schedule of the next before we are like, you know, by when we come to the finishing time, last 2-3 lectures of photosynthesis are left. At that time, I'll give you the schedule for the next lecture or the next chapter. Okay, waiting for the questions in case if you have any doubt here in cyclic and non-cyclic photophosphorylation. If no doubt, then we will come to dark reaction. Telegram channel, yes, I am on that channel. Ma'am, why cytochrome and PC are absent in non-cyclic? See, electron transport chain, it is not that this is how they would be arranged. This is how the membrane proteins are. Now here, if you remember the structure of biomembrane, in biomembrane or plasma membrane, there is a phospholipid bilayer, right? And in this phospholipid bilayer, there were extrinsic and intrinsic proteins. These outer proteins are called extrinsic proteins, and these are the intrinsic proteins. They can be partially or completely embedded. So these proteins, they are acting as the electron transport chain. This is how they are located in the membrane. Okay? So this is how they are present, and basically it is, if you're talking about an electron transport chain, then what exactly it means, that ferredoxin is able to accept the electron which is at a higher energy level. Cytochrome accepts electron which is at a slightly lower energy level. So depending upon their capacity, their arrangements are there in the membrane, and that is where they are working as the electron transport chain. Alright, so if no doubt in this part, then we will start with the dark reaction, that is Kelvin cycle, C3 cycle. There are many names. So we have talked of light reaction, and in light reaction we talked of two pathways, that is non-cyclic and cyclic. Photophosphorylation. This is the first one, and why we are calling it light reaction, because these pathways cannot take place without light. What is the chemical nature of cytochrome? It is an iron copper containing protein. It is a conjugated protein, cytochrome. Okay? Alright, now let us talk about the dark reaction, which is also known as biosynthetic pathway or phase. And we will talk about the scientists and how exactly we know the detail of this dark reaction, and then we will actually draw the dark reaction. Melvin Kelvin was the scientist who actually worked using carbon dioxide with C14. The alga used was chlorella to study the path of carbohydrate synthesis during photosynthesis. What was done by Kelvin? Kelvin took a beaker. In the beaker there was water which was placed, and in this water was the green alga chlorella. So chlorella, green alga, that means it is going to perform photosynthesis, and this setup was exposed to sunlight. So there was sunlight which was falling on this complete setup. Because this chlorella is a green alga, it must have started performing photosynthesis. And what Kelvin and his co-workers did, after every few seconds they would take few chlorella cells and dip them in hot alcohol. So say some chlorella cells were taken and dipped in this after 1 minute, and now because they were dipped in hot alcohol, the cells died, and then the cells were analyzed, which chemical compound is detected in them. Then after 1 minute this was done. After 2 minutes the cells were taken, they were dipped in again hot alcohol. So this was done every minute, every few seconds. Cells were taken out from this setup and the cells were killed, and the chemicals which were synthesized in those cells were studied, and that is how we know that the first compound is A, then the second compound is B, then the third compound is C. So the entire pathway was given by Melvin Kelvin, and he used, see, if carbon compound is formed, then you need to know that which new carbon compound is formed. So how are you going to trace or track that? You take C14. So this is how it was actually calculated. Dark reaction is known as C3 cycle. The reason is the first stable compound formed is a 3-carbon compound. And how do we know? Exactly same method by which Kelvin found it out. So when Kelvin was trying to perform his experiments, he found that there was a compound which was a 6-carbon compound, but it immediately got dissociated. So he could never isolate that compound. So first stable compound which was isolated was a 3-carbon compound, and that is why it is also known as C3 cycle. Named after scientist, it is also known as Kelvin cycle. So there are various names given to this pathway. It is called dark reaction. Why is it called dark reaction? Because it is not dependent on light. So you can call it light independent reaction. See, it is light independent reaction. Where does it take place? It takes place in stroma, because stroma has all enzymes required for C3 cycle, and that is why dark reaction takes place in stroma. ATP and the reducing power which are synthesized during light reaction are utilized in this particular phase, and that is why we said that in light reaction non-cyclic photophosphorylation is the major pathway, because for dark reaction to take place, both the things are required. ATP is also required and NADPH2 is also required. Okay, it is NADPH2. Dark reaction is completed in 3 major steps. First is carboxylation, then is reduction, and then regeneration of RUBP. So if this basic thing is clear, then we will come to the actual pathway. Carboxylation is addition of carbon dioxide to something. Addition of carbon dioxide, and this carbon dioxide is getting added to RUBP. Addition of carbon dioxide to ribulose biphosphate. Its abbreviation is RUBP. Then there is going to be a redox reaction, and it is also known as reversal of glycolysis, because in glycolysis there is a particular step, and the reverse of that step is actually the redox reaction which takes place. And then this ribulose biphosphate which is used has to be regenerated. That is the third step. So in these 3 steps, dark reaction, 3 major steps, dark reaction will be completed. Rubisco fixes carbon dioxide in C3 cycle. Sanjay is asking this. Rubisco is an enzyme which helps in carboxylation of RUBP in C3 as well as in C4, but the location is different. Once we finish C4 cycle, then we will have a comparison between C3 and C4. So that time we will be able to understand why this location is different. Okay, so now we are on this pathway which we will be actually drawing all the steps. Remember, the first step is carboxylation. So carbon dioxide is to be accepted or added. So carbon dioxide is accepted by RUBP, ribulose biphosphate. This is carboxylation. But for this reaction to take place, an enzyme is required. And this enzyme is Rubisco. So here we will write down about Rubisco. Rubisco normally acts like carboxylase. So it is RUBP carboxylase oxygenase. What does this mean? It means that Rubisco acts as carboxylase as well as oxygenase. So here we will write that it acts as carboxylase. What would be the function of carboxylase? That is addition of carbon dioxide. But in some conditions, Rubisco acts as oxygenase. What are those conditions? If temperature is high and if oxygen concentration is high and carbon dioxide concentration is low. In these conditions, Rubisco is going to act as oxygenase, and that is why this word oxygenase we have written in bracket, and the main function, that is carboxylation, is written separately. So generally it acts as carboxylase, but if the conditions are any of these or both, that is if the temperature is high, oxygen concentration is high, carbon dioxide concentration is low, in that case that Rubisco is going to act as oxygenase. So when it is carboxylase, what is it doing? It is helping in addition of carbon dioxide, and when it acts as oxygenase, it will help in addition of oxygen, and that is what is going to create problems. Alright, so now we get the first stable compound, and the first stable compound is a 3-carbon compound. And because of this, this is called the C3 cycle. It is P-phosphoglyceric acid or phosphoglycerate. It has a phosphate, and that phosphate is on the 3rd carbon, so it is known as 3-PGA. Okay, what has happened so far is only carboxylation, addition of carbon dioxide to RUBP. Now we want to track carbon numbers. So what we will do is we will write down the carbon numbers, and to make this cycle easier for us, we will start with 3 carbons. So if there are 3 carbon dioxides, they will be accepted by 3 RUBPs. Let us keep a track of carbons. So carbon dioxide is 1 carbon, so 3 into 1, that is here we are bringing 3 carbons. Okay, RUBP is a 5 carbon compound. So RUBP is a 5 carbon compound, and there are 3 molecules. So total there are 15 carbon atoms which we are talking about. Okay, so 15 carbons are coming from RUBP and 3 from carbon dioxide. You will realize why we started with 3. We can start with 1, we can start with 3, we can do it with 6, but it is easier for us to understand. Once we do the cycle, you will realize that if we would have started with 1 carbon, then we would have encountered some kind of difficult steps. Now, how many carbons so far? 15 from RUBP and 3 from carbon dioxide, so total 18. PGA is a 3 carbon compound, and you need total 18. So there will be 6 molecules of PGA. So 6 into 3 carbon compound, 18 carbons. So are we able to keep a track of this carbon? This is step number 1, addition of carbon dioxide. And which molecule accepts this carbon dioxide? That is RUBP. The enzyme is Rubisco. Normally, when we talk about the normal process, Rubisco is acting as carboxylase. Step number 1 is complete. Now there is step number 2. The second step is redox, reduction, and we know redox is oxidation. Something is going to get oxidized and something is going to get reduced. Here we would need the reducing power, because it's a redox reaction. And where is that reducing power? The reducing power was synthesized in light reaction, and that was NADPH2. So now NADPH2 is going to give that hydrogen. It is going to reduce something. So what is that thing which is going to be reduced? That is PGA, and PGA gets reduced into PGAL, aldehyde. So this PGA is phosphoglycerate, and PGAL is phosphoglyceraldehyde. So acid is getting reduced into aldehyde and the phosphate is on the third carbon. This is also a 3-carbon compound. Phosphoglyceric acid is also 3-carbon compound and phosphoglyceraldehyde is also 3-carbon compound. So what is that, or which is that molecule which got reduced? It is acid into aldehyde. And what is oxidized is NADPH2. So we get now NADP. This is redox. So one molecule is getting oxidized, the other is getting reduced. And this step is reverse of glycolysis. What happens in glycolysis is phosphoglyceraldehyde is obtained and that phosphoglyceraldehyde changes into phosphoglycerate. So here acid is getting reduced into aldehyde. In respiration, aldehyde is oxidized into acid. So when we talk of respiration, glycolysis, we will talk about this also. Okay, so second step is done. Let us keep a track of this carbon numbers. Let us come to this carbon which we were talking about. So 3-PGAL aldehyde is synthesized, which is again a 3-carbon compound. So 6 molecules of this, and again we have 18 carbons, so we are still maintaining that carbon number. Now what happens is the third step. This PGAL has to enter 2 pathways now. Some PGAL will help us in giving carbohydrate and some PGAL will help in giving the RUBP molecule. That means regeneration will take place. So from here, this PGAL, one molecule of PGAL follows a different path and the other, that is 2 molecules, out of 6, 1 molecule of 3-PGAL gets separated, that is 3 carbons here, and 5 molecules of 3-PGAL follow the different pathway. Let us keep a track of this carbon. Let us see what is happening to our carbon numbers. Total there were 6 molecules of 3-PGAL, right? And that is how we have 18 carbons. Now 1 PGAL gets separated from this cycle and follows a different path. So how many carbons have been separated? 3 carbons. The PGAL molecules which are still in the cycle and helping in regeneration, they are 5 molecules. So 5 molecules of 3-carbon compound, how many carbons? 15 carbons. And these 15 carbons would help us in generating RUBP. Okay? Now, when this redox reaction is taking place, reducing power is required. Don't you think energy is also required, and how many of these reducing power molecules would be required? So to reduce 1 PGA into PGAL, 1 is required. We have how many? 6. So there would be 6 NADPH2 getting oxidized into 6 NADP. And in this particular pathway, ATP is also required. Energy, because old bonds are to be broken, new bonds are to be made. So here 6 ATPs will be required and we will get 6 ADPs. Okay, so far what has happened is step number 1. This is our step number 1, that is carboxylation, addition of carbon dioxide to RUBP in presence of Rubisco. The second step is redox reaction or reduction. Here reducing power is required and ATP is also required. And then PGAL, phosphoglyceraldehyde, which is obtained, they will separate into two pathways. Some molecules will help in synthesis of glucose and some will help in regeneration of RUBP. So here we have started with 3 carbon dioxides, okay, and we are getting 6 molecules of PGAL. So if you have 6 molecules of PGAL, you can separate 1 molecule here and 5 molecules in the regeneration step. Imagine if we would have started with 1 carbon dioxide, then here the molecules would have been in fractions, and that actually complicates everything, and that is why we start with 3. We can start straight away with 6 also. Now 3 carbons are separated here. If one more such cycle takes place, then how many PGAL will get separated? 2 PGAL, that means 6 carbons. So this separated PGAL is going to help in glucose synthesis. Now we have 5 PGAL which are helping in regeneration of RUBP. Now this RUBP, each molecule has by 2 phosphates. So how would that phosphate be taken or obtained? Again, ATP will be required here and we will get ADP. So to regenerate 3 RUBPs, 3 ATPs will be required and 3 ADPs will be obtained. So this is our step number 3, that is regeneration of RUBP. So step number 1 is carboxylation, step number 2 is reduction, step number 3 is regeneration. If we want to synthesize 1 glucose molecule, that is 6 carbons, so can we double it, everything? So that will give us, like, to synthesize 1 glucose molecule, how many ATP and NADPH2s will be required. So we are just going to double everything. I'm going to write 6 carbon dioxides here, 6 RUBPs would be here, 12 molecules of PGAs will be, everything has to be just doubled. 6, that is 12 NADPH2s would be required. 6, if there are 6, we'll have to write 12 NADP will be generated. Here 12 PGALs will be produced. Here 2 PGAL will separate and 2 PGAL will bring 6 carbons. Then 10 PGAL will help in regeneration and ultimately we will get 6. So this will be doubled, this will be doubled, this will be doubled, this will be doubled. So now if you have to count how many ATPs are required to synthesize 1 glucose, you can start with 6 and then you can reach to that number in one go. So here there are 12 NADPH2. This is requirement for synthesis of 1 glucose molecule. One thing is 12 NADP here, this 12. And how many ATPs are required? 12 here and 6 here, that is 18 ATPs. This is very, very, very important. This is what is required to synthesize 1 glucose molecule through Kelvin cycle. But this number 3, that is 3 carbon dioxide or 6 carbon dioxide, we study the cycle by using 3 carbon dioxides or 6 carbon dioxides to make our life simple. Because if we start with 1 carbon dioxide, here the fractions are to be written, because only 1 part goes here and 5 parts would help in regeneration. So that fraction thing makes the whole cycle difficult, and that is why we either start with 3 carbons as carbon dioxide or 6. That is our choice. But the most important statement which you have to write down on the page where you have drawn this cycle is 6 Kelvin cycles are required to synthesize 1 glucose molecule. That means it is always 1 carbon dioxide which is accepted. Why did we change it to 3 or 6? Is only to make this whole thing easy for us to understand. Otherwise, it is always 6 Kelvin cycles required to synthesize 1 glucose molecule. Because if you remember, when we talked about dark reaction in the beginning, we said there are 3 major steps. Actually, there are many intermediate steps which are there. Now, 3 steps, a quick recap of this. Dark reaction is basically reduction of carbon dioxide to obtain carbohydrate. So carbon dioxide is accepted. First step is carboxylation. It is accepted by RUBP and we call it carboxylation of RUBP. This is done in presence of an enzyme, that is Rubisco. And Rubisco is a protein which is the most abundant protein in the living system. So Rubisco is most abundant protein in living system. In the entire living world, this is the maximum protein which is found, that is Rubisco. Rubisco changes its behavior if conditions change. If temperature increases, if oxygen concentration increases, then Rubisco starts acting as oxygenase and it creates problem. We'll talk about that later. So right now everything is normal. Rubisco is acting as carboxylase. So the first stable compound which is formed is PGA, phosphoglyceric acid. Now second step is reduction. This phosphoglyceric acid is reduced to phosphoglyceraldehyde. For reduction, the reducing power is required and energy is also required. Both these things, ATP and NADPH2, were synthesized in light reaction. This phosphoglyceraldehyde which is synthesized, a part of it goes for glucose synthesis or carbohydrate synthesis and a part of it goes for regeneration of RUBP. For regeneration of RUBP, again ATP would be required. So if we have to sum up the requirement, 12 NADPH2s and 18 ATPs are required to synthesize 1 glucose molecule. And how many Kelvin cycles take place to synthesize 1 glucose molecule? 6 Kelvin cycles. If we say 6 Kelvin cycle, that means in every Kelvin cycle there is only 1 carbon dioxide molecule which would be taken in. So this is our dark reaction, C3 cycle or Kelvin cycle or biosynthetic phase. Why are we calling it biosynthetic? Because here we are able to synthesize that biomolecule, that is carbohydrate or glucose. Do you have any doubt in this particular part? If yes, you can message that thing and then we will See, if there's any confusion here, we'll talk about that. So I'm waiting for your doubts. You can ask questions on everything, non-cyclic, cyclic and dark reaction, all three. Rubisco fixes carbon dioxide in C3 cycle. Sanjay, it is like in C3 as well as in C4. I told you, I think in the beginning, that the location is going to change in C4. So with this dark reaction, actually photosynthesis is completed. Photosynthesis is when plants use carbon dioxide and water to synthesize their own food in the form of carbohydrate, and oxygen is given out as a byproduct. That is our process of photosynthesis. So this is our carbohydrate which is synthesized, and it is synthesized in the form of glucose. Okay, any doubt so far? So I guess there is no doubt in this dark reaction or this Kelvin cycle. What I would suggest is go over the pathways in detail, go over all the steps and keep a track of carbon numbers. And if you're able to keep a track of carbon numbers, the cycle becomes easy to understand. Steps of Kelvin cycle we have written in short. We have written down those. One is carboxylation, then was redox, and third is regeneration of RUBP. It is carboxylation is addition of carbon dioxide to RUBP in presence of Rubisco. Redox is reduction of phosphoglyceric acid to PGAL, and oxidation of NADPH2 to NADP, and regeneration of RUBP is regeneration. Now, what is required for Kelvin cycle to take place? Requirement is ATP, requirement is the reducing power. Both these things are synthesized during light reaction. Both these things, and obviously carbon dioxide is required, which is to be reduced. Okay, I got one message from Rahul who says no doubt ma'am, that is very good. I assume that others also don't have any doubt on this, and the initial things which I told you about the subscription, so don't forget to subscribe, and subscribe as early as you can, because the prices are going to change very soon, and use the code so that you are able to get 10% discount. So I will see you in the next lecture, and we will continue beyond that. But before we start the other things, what we will do is I'll wait for your doubts for a few minutes. So revise light reaction, both the pathways, dark reaction, complete, draw it once so that you're comfortable with it. Okay, so I'll see you in the next class. Till then, take care. Bye bye.